so she can pass it.
Welcome to the Geospatial Forum. It's a pleasure to have Brian Rich here with us today. He's an associate professor in the Department of Statistics right here at NC State, so he's a good colleague for all of us. Brian received his PhD. Wow, look at fanfare. If your phone has to ring in a crowd, that's what. He was competing with the bottom. Now my phone's. Brian received his PhD from the University of Minnesota and then started as an assistant professor here at NC State in 2008. He's doing all kinds of cool things in statistics at very advanced levels, but one really great thing that I've seen about Brian since I've been here is he's very collaborative, and he's working with people across a lot of different disciplines. I think mostly trying to get the domain data, but it goes that the knowledge transfer goes back and forth to do really exciting research. With that said, we recently invited Brian to be a faculty fellow in the Center for Geospatial Analytics, and he just started, so he's a member of the family. So we're really excited about that as well. So no further ado, it's great to have you here, Brian.
Thanks a lot, Ross. Thanks you all for coming. This is great. So as Ross mentions, I worked on a lot of things. That's one of the great things about being a statistician is you get to work on something different every single day. Most of my work is in spatial statistics. I'm a Bayesian spatial statistician, I guess. I'm a fairly applied statistician. Most of my work is on more environmentally related work like air pollution, climatology, things like that. This is a bit of an outlier if you look at my research portfolio, but it's a super cool project, so I want to talk about it with you guys. This is mostly the work of Neil Grantham, who is right there. So this is one of the chapters from his PhD thesis. This is also joint work with Eric Laber, who's an associate professor in the statistics department. And then many of you probably also know Rob Dunn. So Rob provided the data and sort of the big picture plan for some of this work. And all of the lab work has been done by Noah, who's from Colorado. So this is definitely a very collaborative team. We could not do this, any one of us individually. It really takes all these pieces to make it happen. Many of you probably know what a microbiome is more than I do, but let me describe it as best I can. Microbiome is, the people we work with are mostly ecologists working on microbiome. And so what they're interested in is the different species of fungi and bacteria, how they relate to each other, how they relate to different factors in the environment. And so for a particular sample that you might take, say, in a piece of dust, which is what we'll do, it might include 30,000 species of fungi, tens of thousands of species of bacteria. So for an ecologist, this is really exciting because if you study, say, birds or amphibians or something, you take one sample, you might get 3 or 4 different kinds of birds, but here we can get a whole suite of species. And this is actually pretty affordable now. Just taking a dust sample, the way these data were collected, they took a sample of dirt, they mailed it off to Colorado, Noah ran it through a genetics algorithm, and it spit out all of this data about the presence or absence of all these species for dozens of dollars. And so this is something now that you can do on a large scale. You can take many samples, you can see how the microbiome varies over space and time and with different ecological conditions. And so there's a lot of interest in this work. Most people, when they think of microbiome, they think of gut microbiome, and that's certainly important. And we have some of that data for these samples, but that's not what I'm going to focus on now. There's a lot of other areas. Agriculture is another thing that Neil is working on. We're going to today talk about using microbiome for forensics. Microbiome is a big deal in science. I think it might be one of the next big things, if it's not already a big thing. So the White House recently has launched an initiative to try to get more people involved in microbiome research. A few of the things that are flagged in this initiative was collaboration, better tools, and recruitment. So collaboration and better tools are certainly music to my ears as somebody who develops statistical algorithms for a career. It's also a really great place to be if you're a statistician or a geospatial analyst, because these are not simple problems. They're super high dimensional, tens of thousands of species. There's a lot of challenges working with the data in terms of false negatives, false positives. It's really sparse. So in most samples, there are, of the species that we've collected, there's something like 5–10% of them show up in one given sample. So if you just look at the abundance of each species, you see tons of zeros. For the species that are present, they have super large counts. So you get these extremes of lots of zeros, lots of extreme values. That is certainly interesting to deal with as a statistician. And then something we haven't explored a lot in this work, but we have in other work, is the complex dependence structure. So there's 30,000 species. Some of the species fill specific ecological niches. There's some interaction between them just that we'd like to study. It's really hard to understand how 30,000 species interact with each other in any meaningful way. So I think there's a lot of exciting opportunities in this field. So now let me describe the specific data set that we're going to use. I guess I mentioned some of this. So Rob Dunn runs a citizen science project called Wildlife of Your Homes. And if you're interested in this talk and these data, a lot of this data is now publicly available on his website. So to collect these data, he got, now I think it's up to 12 or 1,500 people from around the US. So these are citizen scientists. As I mentioned, they took a scoop of dust from their house. Actually, they did this in a few places. They took one inside the house, one outside the house. And then inside the house, they took a few different things like pillowcases and a few different samples like that. These people were incredibly generous with their time and information. They recorded a lot about themselves and their home. They recorded if they lived in an apartment or not, if they had air conditioning or not, if they had a cat or not. We were able to get things like their land use in the neighborhood. They recorded whether they did or did not use several pesticides. They provided health information. So it's an amazing data set. We're only going to use a small part of it here. From these samples, as I mentioned then, they took these dust samples. NOAA in Colorado processed them. It revealed which species of fungi and bacteria were in the sample. We're not going to use bacteria in this one, but just restricting our attention to fungi alone, there were 50,000 species of fungi that we're going to use. Guys have any questions about this? I literally do have 30 minutes prepared. That's what John asked, so I have plenty of time for questions. So if anything's unclear, let me know.
Were these people paid anything at all, like for their time, or any kind of like gift card or anything like that, or?
It was volunteer primarily. I don't know if they received a gift card. Do you know this?
No, they receive access to the data afterwards, so they can do their stuff with it.
Yeah. That was your comment?
That was my question, is what level data they got to see about their own house and how creepy it was.
They got all of it.
You have to get over thinking that fungi is creepy. Fungi is helpful. It took us a while to get in that mindset, too.
Is this an open data set?
Yeah, sure.
Who asked that? Harder.
We can access this data.
You can access most of it publicly. I don't know if all of it's...
Some of it's scrubbed for confidentiality.
Yeah. But all of this data... Okay. All right.
Also, what is the spatial granularity of the data?
We have latitude and longitude down to 2 significant digits. So, pretty good, I think. For our purpose, we're going to try to predict where the sample came from. We cannot predict at that resolution. So for our purpose, it's good enough. Anything else? These are great. Keep them coming. I really only have 30 minutes, so. Okay. So there's dozens of papers written with this data set, as you can imagine. Given that microbiome analysis is so new and this data is so cool, there's a lot you can do. So we're going to predict or take this one specific problem. So this is related to forensic science. So the research question is, I give you a dust sample. From that dust sample, you figure out which species of fungi are in this sample. And then the question is, where did that sample come from? So we would like to take in as inputs to the statistical algorithm is the vector of presence-absence of these 30,000 species of fungi. What we would like to spit out is the latitude of longitude where the sample came from and a 95% prediction region. Okay, so that's the plan. In a little bit of mathematical notation, this is what that translates to. So X is the covariates. Here, that's a vector of length 30,000 with zeros and ones, whether the species are absent or not.
Sorry. Is it really a good intro? I just was, are you interested, you're interested in geography, but then are you just looking at the outside samples for this? Or?
Yeah. These are just the outside samples. Yeah.
Thanks.
Yep. Okay. Okay. X is long. It's zeros and ones. S is a latitude, longitude. Okay, so this is our first crack at this. So this paper appeared in 2015. I think this picture summarizes it pretty well. This pointer, it works on the wall, but not the screen, so I'm not sure how useful that's going to be. But just in pictures, here's what we did for this simple algorithm. So the focus of the talk is what we did after this, but this is sort of going to serve as our baseline. All right, so here's organizing the data. They came from N homes. There are M species of fungi. These are the long vectors X of zeros and ones about whether each species was at that house or not. We took each species, smoothed them out with a pretty straightforward spatial smoother. I'm sure you guys could do better than this, but this is what we did. Okay, and then we do this for each species. So this gives us 30,000 maps of where each species tends to live. And then we have an unknown sample. We don't know where it came from, but we do know which species of fungi were from that location. And so you try to piece this together. This sample had this one. That probably means it's in the southeast. It did not have this one. That means it's probably not in the north. It did not have this one. That means it's not in the west. And you start to piece all this information together. You overlay all this maps. You look for where there's an intersection. And then that produces a predictive distribution. Formalizing that a little bit, you can call this discriminant analysis or Bayesian discriminant analysis. Okay, so then, like I said, laying all these maps together, going to Bayes' rule, you get the predictive distribution, and then this is what you get. So you get our best guess as to where the sample came from, and then these are 50, 90, and 50, 75, and 90% prediction intervals for that sample. Any questions about that? Okay, easy peasy. So then here's some metrics of how well this works. This is one particular sample and its prediction error. Its error is 153 kilometers, which turns out to be about the typical error, which is why Neil picked this one to present. So this one, it was really from whatever this is, Northern Virginia. We predicted it was going to be Central Virginia. We missed by 153 kilometers. You apply this, so we did leave one out cross-validation, so we fit the model to n-1 of the points, made a prediction for that point, and then redid this a whole bunch of times. This gave us a bunch of prediction errors. The median prediction error was 239 kilometers. Some we got to 4, one we missed by 4,000, so there's still, we can, certainly room for improvement. But, 239 kilometers, that's actually better than I was expecting. I don't know why I was expecting it to be large, but I was pretty surprised when it was that small. Okay. And this is super cool. So after we published this paper, these guys called us up, and they had a CSI Cyber episode related to this. So in this episode, somehow they needed to take a laptop, and they needed to have some sciencey words to tell them that this laptop came from a horse farm in Houston, Texas. So they asked us for a few fungi and a kind of a believable story that could do it. And I don't know anything about horses or Texas or science or anything, but we were able to come up with something that made them happy. Okay. So anyway, so that was pretty cool. What's that?
Where's the clip?
We have a clip, actually. We couldn't figure out how to get it in the slides. I don't know.
Yeah, we legally put it in the slides.
We legally put it in the slides. That's right. Yeah.
How did you put that on your CV?
We were just talking about how to make sure people get credit for really important scientific research. So yeah, we got to think of a way to do it. Okay. All right, so that was our first approach. It works pretty well. I think it's kind of a cool application. But that's not going to get Neil a PhD in statistics. So we tried to go a little bit beyond that. So for a few reasons why that is not the optimal way to go. One thing is that we ignored the dependence between species. So we just took those 30,000 maps. We assumed all species were independent. We laid them on top of each other, weighted them all the same, and just took what we got. They are clearly not independent, and so we could try to account for that. So if instead of just making all the maps laying on top of each other, if you tried to estimate the whole distribution between all 30,000, so which parts of the country each one lives in, and then the, say, joint probability of them showing up, something like the correlation between them all, that would be better, but my gosh, that's hard, right? Because there's a 30,000 by 30,000 covariance matrix to estimate, and we can't do that. So we decided to take a different direction. We decided to focus more on statistical models that are popular in the spatial statistics literature. So when you think about the response variable being a latitude and longitude, then that is spatial point pattern theory. So we decided to try to take that information into account. But then it's so high dimensional that just the off-the-shelf things that you see in spatial statistics textbooks, they just don't apply when you have 30,000 predictors. So we tried to use some more machine learning-y things, and I'll show you some deep learning results in a second. So here is spatial point pattern theory. Well, I guess this is not theory, but this is a common model fit to data where the response is a spatial location. Typically what you do is you assume that the data arise from a Poisson process. And then because we think that some locations have more intensity than others, you get a non-homogeneous Poisson process. So this lambda of S, this is high where you expect a lot of points, so a lot of samples to have originated from, and it's low where you don't expect a lot of samples to come from. And so the goal then is to estimate this function, which is a spatial surface, something like a probability density function that tells you the most likely places that samples come from. Okay, and so we want that distribution of points to depend on the covariance x. That's the 30,000 vectors of zeros and ones. So we need a surface that isolates the highest intensity areas, but that also changes with the covariance. Okay, so non-homogeneous Poisson process is one way to do that. If you do that, this is what you get for the likelihood. It's not the most straightforward thing. It's got this integral in there, which is a pain in the butt. We're going to not end up doing this, but this is here to more or less say that just the standard things that people do don't work well here, because you have to take this integral. That's hard. And you also have to come up with a model for how this intensity surface depends on the covariance. So you could try to come up with some sort of linear model or something, but I mean, there's 30,000 of these things, so just writing down a linear model is probably not going to work. Okay, I think I probably said most of this. Oh yeah.
Do you have a good representation across the entire U.S.? A distribution map or from all the samples?
I don't have it here. I think it's in here. I'm going to make you guys dizzy. This is hypothetical stuff. I'll just say it in words. Yes, there's at least one sample in all 50 states, at least 48 states. There's way more in North Carolina than you would expect, even based on just population intensity. But it sort of follows the population, except North Carolina has more, and there's 1,000 points. So it does get sparse in the Dakotas. In Los Angeles and New York, it's pretty fun. Any other questions?
And for the samples that they collected, did they specify which basin side of the house?
Nope. I don't know that I have that. How do you feel that would affect the microbiome distribution?
North facing versus south facing might have some effects.
Maybe. I don't know. I don't know anything about fungi, so I don't know. I guess from our perspective, that probably wouldn't be super helpful because the sample of dust probably wouldn't carry that information with it anyways. And so if we built a model that relied on that, then you need to figure that out before you could apply it. But I don't believe they took that into account. Okay. Okay, so here's our plan. Where was I? Skipped around all over the place. We got to this one.
Okay.
Yep. Okay. All right. So deep learning. Probably most of you have heard of deep learning, I guess. But this is what's really taking off in the machine learning, data science-y community, and I guess the statistics community as well. Where it is good is when you have data that's super high dimensional, like ours, 30,000 predictors, and where there's really complex dependency structures, like that 30,000 by 30,000 covariance matrix I alluded to. It's also super useful when you don't have a lot of scientific information to bring to the table. Like if you had information about a priori which species were correlated with each other, or maybe groups of species that were correlated with each other, you would probably do better by using that. We don't have that. So we just want to dump all these zeros and ones into an algorithm and have it spit out predictions. That's our goal. So we're not interested in learning about the science here. We're just interested in making good predictive model. So this is where deep learning comes into play. But we have to do some modifications of these regular deep learning algorithms to fit it into this specific spatial forensic science application. So our plan was to try to turn this into a classification problem. So a classification problem would be if I have 10 different possible outcomes and I want to take the x's and try to predict which one of these 10 outcomes it came from, that's classification. So deep learning is super good at that. There's dozens of algorithms that can do it. But when you just look at our data, it is not clear what those 10 classes should be. So we artificially created some classes so that we could take these artificially created classes, dump them into a classification algorithm, and let deep learning do its thing. So the way we artificially created these classes for this classification algorithm was to generate a random partition of the spatial domain. OK, so I think I can show you this with pictures instead. I'll get back to this. So we split the region up into capital K intervals and then turn this into a supervised classification problem. Given that we have just K classes now, we can assume that that intensity function, lambda of S, was constant in each region. So if we have a region that is the Dakotas, we don't try to predict within the Dakotas. We just try to predict that it came from the Dakotas. So this then simplifies the problem a lot. We don't have that integral anymore because we just have 20 regions, and we just need to know which region it's in, and we don't have to worry about that. OK, so we can more or less dump this in. There's a million different algorithms that we could have used for this. Neil took this one, deep neural nets with this particular loss function. But I don't know how critical this specific algorithm is to this. This is something we're still working on.
So, I have a question. So you artificially created the divisions, right? So why haven't you tried to cluster it and make some kind of more natural divisions?
If you actually had some divisions that were meaningful to you, you should use those instead. What we're going to do is we're going to do this. So the next thing I was going to say is that we don't really care about these partitions. We just randomly generated them.
Okay.
So what we're going to do is just repeat this a whole bunch of times and sort of average over this randomness in the partitions. OK, so let me finish this slide then. OK, so here's our algorithm. Generate random things, partitions, train a different deep neural net on each one of these random partitions. And then we're going to repeat steps 1 and 2 a bunch of times. So we'll have, because we don't care about any one of the individual partitionings, that's just randomly created by us. So we're going to do this capital N times. And then this is going to be kind of the analogy of overlaying the maps from discriminant analysis. We're going to overlay these different classifiers. And that's how we're going to get classification at the latitude, longitude level rather than this silly partition level. OK, so here's Neil's fancy plots. OK, so the way we create the partitions is we randomly throw these blue points out. So there's capital K of these blue points, and these are just uniform over this grid of points in the US. This is just a Voronoi tessellation based on those points. So the tile that you get assigned to is the one that is closest to the point that is the parent of that tile. Then we throw away the location of the points, and now we've got a tiling of the US. This one has something like 35 or so, 31, 33, looking for the biggest number. Let's say 33 tiles. So now we're going to train a classifier to predict which one of those 33 tiles that it came from. Here's the data that you would use to what's that? This is the data. Sorry, I don't want to give the whole talk away before showing you the data points. But Here's the data points. So you can see North Carolina has a lot. Other than that, yeah, I guess it's pretty sparse up here. But anyways. Okay, so here's the data. This is what you have to train the algorithm. So what you're going to look for is the typical microbiome profile of these 100 samples, and if the new sample kind of matches up with that, you're going to classify it to that tile. Yep.
Is that actually 100% of the data, or are you reserving some for some cross-validation purpose?
I'm so glad Neil came to this talk.
Yeah, this is some data points withheld from this. So yeah, this is 90% of the data.
Good. Okay. All right. So we dump this in to Neil's fancy Python package, and it spits out then a predictive probability that a new sample came from any one of these 35 cells. This one, it has probability of 1 of coming from this one in the northeast and 0 from coming through all the rest. So that one is pretty confident about. But this isn't what we want. We don't want to say that it came from that tile, which means nothing. We want to give a latitude and longitude. We want to put a pin in a map. So we're going to repeat this a bunch of times. Here's another one. Generate random partitions, fit the classifier. These are the probabilities on each of these tiles. So for this one, I don't know, what is this, like 70%, 30%, 5% or something. So it's still convinced it's in the northeast, but it's not sure which tile it came from. Here's another one where it's convinced that it came from that cell for sure. This other one this isn't super useful. We already knew it came from the northeast, so it happened to be a big piece of tile that it could pretty clearly classify it into. Okay, and then here's another one where it's mixed. Okay, so most of the time it's going over there in the northeast, but we're still not quite sure exactly where in the northeast. So what we want to do is average over these different partitions. Here's the way we're going to do it. So this is going to be what we're calling here the geolocation function. I guess this is just the predictive distribution. I give you a map, or x, which is my zeros and ones. My predictive distribution is g. It is take the classifier that comes from the from each of these classification objects, and then average them. And these weights are so Wj is the weight that's given to random partition number j. Okay, and then so this is the whole distribution. So if you wanted a 95% region, you could pluck off the locations that have 95% probability. If you want just the best guess, you would take this function and find the location that maximizes that function. Any questions about this one? All right. A lot of tuning for these things. Honestly, the great thing about neural nets is that they're super flexible. The downside is that they're really hard to tune. I won't go into what all of these things are, but there's 4 or 5 different inputs that you have to put in there that determine things like how deep the network is and how it manages leafs in the network. And it's kind of boring to talk about, to be honest. It doesn't have anything to do with what we're doing, but it is super important to make sure these things are done carefully. So our plan is to eventually average over these things, because our goal is not to say definitively that there should be 4 hidden layers, whatever that means. Our goal is just to get a good predictive model. So that's what we're going to do. We haven't done it yet. One of the most important tuning parameters is how many of those points, how many tiles there were in the partition. So those are generated randomly from 30 to 60. Another thing that you could tune is the weights that you put on each one of these models. We're right now just taking the straight average of them, but that's clearly not the best thing to do. So we're hoping to do something like upweight the ones that seem to have better predictive performance. And then we're fixing these values. I think eventually we'll try to get a little more sophisticated there. Okay, so here's one example. This is what you get. So this is actually kind of cool. I'm surprised how smooth these things look, because this really is just overlaying a bunch of tiles and then looking at their product, or their sum, I guess. But from this, you can't really tell that it was tiled at all, right? So there's, at least in one of the maps, or one of the partitions, it seems to have separated out every pair of points. And so it looks like a continuous surface, which is what you want. So this is probably, I'm guessing Neil made this a 95% prediction region, and this is the best point. Okay, so in this one, we missed by 96 kilometers. Here's a few more. So here's this sample we're pretty confident came from the northwest, and we missed by 90 kilometers. This one, we're thinking it came from North Carolina, and we got pretty close. It's 53 kilometers. This one, missed by 6 kilometers. I was pretty lucky, I guess. I'm sure there's still some big ones. Neil, did you put any big ones in here? Nope.
Yep.
Okay. Here we missed by 116. Okay, well, anyway, so I only showed you the good ones. The last one is something like the median, though. So here's the distribution. The biggest one is still really big, 2,000, so we missed by several states on one of them. But you can see by doing this more sophisticated statistical algorithm, we were able to divide the prediction error almost in half in terms of the median and the mean. So it's quite a big improvement. Yep?
So you're oversampling in North Carolina. Did you get higher hit rates in North Carolina? Are you having lower errors there?
We have looked into that. We tried to account for that in our classifier by downweighting those samples. So we actually tried to account for that in each state by weighting the sample in the training, but based on the population to sample ratio. So we tried to account for that.
You're doing that by weighting?
In the training, yeah.
Training. Okay.
Yep. Certainly a good question now.
Otherwise, are there any other covariates related to the air, climate, things like that?
Very prescient. It's coming up.
It's not.
I will say when we started weighting, our prediction error got worse. So cheating and knowing that a lot of the samples came from North Carolina was helping us until we corrected for that.
Are there any places where you actually get very similar characteristics of the biomes? So it's hard to actually, if the West Coast and East Coast would be in some areas the same, how would it?
So we have probably the most jumbled map in the history of statistical journals, but we have this map that has each error. So you can see if we predicted here, it should have come from there. And so you can kind of see the areas that are similar there. And I remember it was really complicated, but I don't remember what the answer was. I think it was most of the whole eastern coast was hard. Is that right?
Yeah, it's like areas along the Appalachian Mountain Range are a little bit difficult to pin down, or very, very urban areas. So like in the heart of San Francisco and the heart of New York, sometimes the algorithm gets confused, likely because it's not exposed to definitive fungi species. It's just all urban. So we do have those situations.
Yeah. So we were thinking that that map might guide future sampling, which we haven't done, but that's certainly something we could do. Any other questions? Oh, yeah?
So I'm curious.
Plenty of seats in here, I think.
I'm just curious how this compares to Adrian Bentley's pixel logistic regression, when he actually does present 0-1 analysis of the presence or absence states, where predictive probabilities could be easily having a logic transform.
Yeah, so let me repeat the question for everybody over here. So he was asking how this would relate to a specific statistical model that modeled the presence and absence of each species as a spatial function and tried to make those maps, I guess, a little bit better. Well, there's 30,000 of them, so that's going to be super slow. I do have a student working on that now. I don't think she's working on it for this classification problem because when you try to actually model the individual species, it seems like overkill because probably of those 30,000, 29,000 are just completely useless, and then you've just wasted a lot of time to do it. So we've tried to turn it around where you don't actually have to model the individual species and get that whole distribution of the species and how they relate to each other right. You just put those as covariates and then dump them into some algorithm. But we are trying to work on that. She's calling her project the Microbiome Atlas, that she wants to come up with a whole book of maps about microbiomes and test for ecological covariates for each one of them.
Because the sampling-based approach is very similar to the one that AI for
Okay, well, we can talk about it later, but we're not trying to actually predict 0-1, is a species going to be present there? We're trying to predict where the sample came from. So it's a different problem, but certainly both interesting.
Sure.
Okay. Any other questions?
You mentioned, I think, that they asked people about their use of pesticides.
Yep.
Yeah. Did you look at that throwing off your accuracy?
We have a table of errors that was broken down by different types of houses. So we didn't use it in our model at all. We have looked at which types of houses have the bigger errors. I don't remember how that one related. I don't remember. It's something we've looked at, but I don't remember the results.
seems like it would be kind of a dead zone or something that could be anywhere.
Yeah. So like you're saying, if they use a certain pesticide, then it just kills off certain species, and so everything that uses pesticides is not going to have this set of pesticide-sensitive species. Yeah, that's a good thought.
Well, if they're pulling it off the houses, then it may be affected by the cleaners they use for structure.
Yeah, these are all things. It's certainly messy data, so we have not used any of that.
Just in the residential landscape literature, it seems like a lot of people aren't directly in control of all of their landscaping decisions, so I think self-reporting would be very unreliable.
Okay. All right, I'm almost done. What we're currently working on is we're trying to take this from just the US to something that's international. So we've, a while back, we got funded by the Department of Defense to extend this idea to an international study. So the sampling is currently underway. The objective is to get 20 samples from each of 30 countries around the world. I'll show you a list of those countries in a second. We also said that we would deliver to them some interactive software that implements this method for people in the field. So Neil is working on this. Super cool, super fun. I could never do any of it. It's really neat. Here's the locations where we're going to get samples. I think the European samples, we have some of those coming in already. I don't know that we have the other ones, but within a few months we should have samples from the whole world. We do not know if the prediction is going to be easier or more difficult. Certainly there's a bigger difference between Uganda and, say, Kuwait than there is between North Carolina and South Carolina. That sounds great, but we don't have nearly the sampling intensity, so I don't know if we'll be able to exploit that or not. Yep.
How were those countries selected? Was the DoD selected those, or did you guys come up with those names?
I believe that was mostly DoD driven.
Because I don't see any Western Europe countries.
Huh?
I don't see any Western Europe countries. Why do you think?
We actually do have samples from Western Europe now. They weren't collected as part of this, but independently from that. So these are more sensitive countries, I guess. So they're probably less interested in France versus Belgium than they are in some of these.
Yeah.
Yep.
Yeah, can you say, I mean, you said it's not as dense sampling, but what's the general sampling plan? Like how, I mean, it's not citizen science again, it's like a...
Yeah, no, it's not citizen science this time. They actually have people out in the field collecting these samples. There's a private company that's supposed to collect these. I forget the name of the company. You probably wouldn't know it anyway, so I guess it doesn't matter. But it's not citizen science. It's also not random either. It's still convenient sampling. It's where they are, where they can get samples without putting themselves in harm's way. Where they take a sample and how it's processed is a little more homogeneous. So they're not going inside and outside of houses. It's more like streets and park benches and things like that.
Do you know, like, I guess you'll know as it comes in, proportion urban versus rural convenience, or is there some plan to?
We had discussed that. I don't remember what we decided. We'll certainly know that.
Yeah.
I don't know exactly what proportion will be urban versus rural, but this is information that we'll have. So we'll have lat longs for all of these samples, exactly.
Yeah. I'm interested in like your inability to tell urban spaces apart, because there's in urban ecology kind of urban homogenization hypothesis. So it's kind of cool to see that's kind of playing out even in the microbiome and see how that works out globally. It's kind of interesting.
The Walmartization.
Yeah.
Well, there's some just mixing of dust too, just due to wind and so forth. But
right.
Yeah, that.
I'm kind of curious. Do you have a time of the growing season?
A time of
The collection dates for each of the dust samples. Is that?
We do have collection dates for the dust sample. We are not using it. I imagine it's not winter in Minnesota, but I'm not sure.
Perhaps season matters.
We have asked them, and they seem to think it does not, that it's not important. We haven't tested it, and I don't know if they know better than I do. Yep.
So, I guess among all these countries, only one is island nation. So, thinking about whether it would be interesting to compare like a land kind of continental, how to say it, but countries that are like on the continents versus
like, completely isolated, I guess.
Yeah.
Yeah, I don't know. That'd be great.
Sure. With other countries, I guess.
Yeah, I mean, I imagine Japan, it's big enough, and that's probably pretty distinct. My guess is, I don't know. That would be my guess, is that you could flag Japan versus not Japan pretty well. But I could be wrong. Yep.
Government from these countries know you're doing this?
I'd love to tell you. I don't know. Yes, I don't know if they know they were doing this. I don't think we could hide it from them, but I don't know if they've been warned or if they would care.
Is there like a thought to like make a model to predict where, say, like a car has been within a certain time period? Like there's take some dust off the car and like, oh, we know it has traveled through these areas.
That's CSI.
Is that?
One thing that we had, so following its whole track would be amazing. It is something we've talked about. We couldn't do it with these data. Obviously, you need several samples followed over time. So that'd be really cool. I don't know if it's possible. It's not something we can do.
But is that the point? Is that what DoD wants? They want to have these records so that they can identify the car coming from Saudi Arabia.
They would want to identify the car coming from Saudi Arabia, but the assumption is that car was always in Saudi Arabia, and then, I don't know, the bumper showed up in some other place, and the question was, did this bumper come from Saudi Arabia? But well, I suppose they would love to have this. They would know that the person drove the car from Saudi Arabia up to Ireland or something, but we certainly can't get that.
Or more likely the shoes.
Yeah, shoes would be cool too. Or if you could say something like it originated in Saudi Arabia, then clearly they went through Heathrow, and then now it's in New York. That'd be really cool. Also far-fetched, but cool. Yeah.
Being to get to the sort of covariates with the errors, I'm just wondering if there's a spatial pattern to the errors that might be linked to some environmentally meaningful things, like how connected certain places, like with regards to the jet stream, and like, you know, are there being dust brought in from other places, from a larger region versus a place that's fairly stagnant, and it's much more localized, and places that have a richer biodiversity of fungal life versus others that have only a few species, and how that might influence the errors.
So we have looked at some of that. As Neil mentioned, the biggest errors, most of our long errors happen along the Appalachians. I think it also does mix. If I remember, New York and San Francisco, for some reason, seem to have, these are the big errors. It's always missing those two. I don't know why that occurs. It would be interesting to apply some covariates and try to figure out why those errors happen. We haven't done that. We also, as maybe Ross was getting at, we haven't put covariates into this model at all. So there's certainly more area for work.
Did you guys make an error map? Like map of all the errors? You know.
Yeah, I've been trying to describe this error map. I guess I'm not being successful, but it was so imagine the US. Every sample where we predicted, we drew an arrow to where it was, and then we overlaid all those arrows. So that's what we're calling the arrow map. And if you look at that arrow map, there are a lot of big ones going up and down the Appalachians, a few of them that went from New York to San Francisco.
But I was talking about possible color coding or changing the hue of the color based on the amount of error that you had for each sample, like, you know, distance error, instead of the arrow itself.
Oh.
I was talking about like a heat map kind of error map that give you a different kind of information.
We have looked at that too, I know. Do you remember what it looked like?
Yeah, I made it. The arrow one actually was a little bit easier to discern what was going on, because the issue with the color coding is, do you place the error at where you predicted the point or where it's from, and it got all kind of jumbled. So with the arrows, you can kind of see these kind of different patterns, like especially in the southeast within like South Carolina and North Carolina and Georgia. There's kind of like a patch where they get kind of confused and stuff like that. So you can kind of begin to see different patterns in what are groupings, I guess.
Okay, open questions. We thought a little bit about how to do this global sampling, but I imagine we'll learn more as we go. We have been thinking about combining, I've only shown you results for fungi. We have also tried bacteria for these data, and it was worse. I think we tried combining fungi and bacteria, and that didn't give us any benefit. It just slowed things down. So we've tried that. Another open statistical problem is to, rather than just make a predictive map, come up with a hypothesis test. So if they have a specific hypothesis in mind, and the question is not where did this come from, but did this sample come from Iraq, how to do that differently, and how to do it in a way that could be easily justified and maybe isn't a black box, but something that's simple enough that you could explain to, say, a jury or a judge or something. We're still working on that. Okay, if you guys have any questions, well, you already did have many questions, which is great. I filled the whole 50 minutes. Thanks to you guys. If there's anything else, here's my email and my website. So thanks.
So at the department, have you perceived it as a problem that you actually don't know what's going on there? Because that's what it's commonly said, like you basically don't know what the algorithm is doing inside. Is that a problem for the...
For the sake of prediction, I guess it wasn't our main goal, and so I don't lose sleep over that. I imagine where it would be detrimental is if you had some scientific knowledge that you wanted to get into the predictive model somehow. We don't currently have a way to do that. And if your goal was not to do prediction, but to really understand the geography of the microbiome, we probably haven't contributed a lot to that question either. So for other purposes, it would bother me. For this one, I'm okay with it.
You guys have resources for the work that was done, like a GitHub repository or somewhere that we could reference to see something more about how you guys actually did the code?
What do you have on GitHub? I know we have the...
Yeah, the first project that we described, it's on my GitHub. It's just called Fungi Identify. We're working on putting this new stuff up there when we get a little bit all of our ducks in a row. Yeah.
Any other questions?
How long does it take actually to conclude it?
It's not fast.
Evaluating examples pretty fast. Training into these networks, we train a bunch of them. So each of them, I don't know, takes a few minutes. So it's not the end of the world, but we make tons of them, and then just kind of go from there. So evaluation is fast. Training them is a pain.
The nice thing about this partitioning algorithm is you can do 50 partitions and then fit the models simultaneously in parallel, and so you can get through a fair number of them, but it's not like an instantaneous thing.
Yeah, but it doesn't take days.
I actually thought it did take days. I guess not.
No.
Well, it would take me days.
It's convenient to let you think it took days. Breathing
I think there might be some elevational component to the difference in fungi. Just thinking about that Appalachian correlates to the urination. There may be a certain elevation that you look, and then you get above that, it breaks, and then you have some differentiation.
We have looked at elevation. I think we found that more important was forest versus not forest land cover, which I guess maybe that's confounded with elevation in that area. But there's lots of, I mean, they're writing dozens and dozens of papers about this with these data.
What do you envision as the state of the art for this sort of research 10 years or so? Like down the line, where do you see this going? How long before a police department can have this tool on their laptops?
Like in TV shows.
I don't think it's impossible to do that. I mean, hopefully we'll have something done in a year. We said we would have something done in a year. We will have something that people can put on their laptops. So I don't think that's a limitation. The limitation is just getting sufficient data. If you were, say, the Baltimore City Police Department, you could probably, if you had the time and money to do this, you could probably do a few hundred samples around Baltimore and get something pretty crude up and running, I would think. I think there is a limit spatially to how well you can possibly do, just because there's just certain mixing in the air, and so there's limitations that way. But in terms of collecting data, it's pretty cheap, and computing, Neil's using all open source software, so I don't think that's a huge limitation.